All right, so I want to show you how to do this lab. We were supposed to do this in class, but due to weather, we're not able to do that. So I just want to show you this. So this is the lab we were going to do in class. You just go to the next assignment here. I'll post this again. This is called the Phenotype Genotype Lab. We're going to just practice what we're learning about these. One of your first tasks is to just define each of these terms, right? There's a couple new ones. Genotype, you know what that is. Phenotype, this is just the alleles that the trait has. This phenotype, the expressed trait of how that trait is expressed in that organism. Homozygous always means same allele, same dominant alleles. Homozygous recessive, same two recessive alleles. Heterozygous is two different alleles for the trait. And mutation is something a little different. So mutation, I encourage you to go look at this in your book. Mutation is a change in DNA. So change in DNA. So we know all of our traits are found in nucleic acids, and the nucleic acid that carries all of our traits is deoxyribonucleic acid, or DNA. And sometimes what happens is they change just through copying and transcription, it's called, and we haven't learned that yet, but we're going to learn it. But what happens is sometimes when it's being copied, if you remember in the cell cycle, DNA has to be copied. Well, sometimes when that happens, there's a change in DNA. So we call that a mutation. And that can be caused, if you remember the cancer lab, it can be caused by being exposed to certain chemicals and it changes your DNA. Sometimes it causes cancer. And sometimes there's just a mutation that changes the trait a little bit. Often, though, when you see a mutation, it causes very adverse things. It usually doesn't result in something that's better. Okay, so that's a mutation. A karyotype is actually a picture of your DNA. Let's see if I can show you this here. It's a picture of DNA. I'm going to have you do an additional lab on this just to mess around with it a little bit and see it. And because we've been talking about all humans have 23 pairs of chromosomes. So if I show you this here, this is there are 23 pairs. Here's your X and Y's. It determines whether you're a male or female. But then maybe go back here. Yeah, I'm going to give you this, make a karyotype. And then I'll even do the printable thing here, but it gives you some information on it. And this gives you a pair with just one of the chromosomes, and then you're going to pair them up accordingly. But a karyotype, they can then look at them and see compare them to each other and determine certain things. But that's a karyotype. It's just a picture of your DNA. And scientists can do that. They can figure out, they can complete that and look at DNA. Okay, that's a karyotype. So in this lab, you're going to look at 4 characteristics. You have Studentus unhappius, and their characteristics, they have eyes that are either googly or beady. They have a leg pattern that's spotted or striped. They have a tail that's straight or curly, and they have a body type that's square. There should be an E on there. And round, I'm not sure what happened to these. It may be when I downloaded it. But I'll look at yours. Yours should say square and round on it. Okay. So we're going to look at these 4 characteristics, just like you did a cross for yourself. We're going to do a cross for this organism here that we're studying. Okay, so here it says this: when Studentus unhappius was examined at Copper River Labs, the following results were obtained. So for this organism, it has googly eyes, the dominant trait. It has striped, which is recessive. It has curly, which is recessive, and a round body, which is dominant. The one thing we're going to remember, if it's dominant, it's heterozygous. So you need to remember that. It's heterozygous. So this is going to be homozygous recessive, the traits. And then this one is going to be homozygous recessive. And then this is going to be dominant. So it has the dominant allele, right? It's HE. So here is your allele. You just need to match up a heterozygous for this trait. For striped, it's L, just 2 homozygous recessives. And you're going to take this genotype that it's given you right here, and you're going to put it right here. And then according to this genotype, or really what it says here, googly eye stripe, curly bar, you're going to put it for the phenotype. So you do that. And then draw a picture. What do you think this thing looks like? So just draw a picture. If you can't do it computer-wise, just bring a picture in on your printed out. And then we're going to answer a few questions. Okay, or you're going to cross it. Okay, so you're then going to cross the traits of this thing right here. You're going to write all these traits, right? And then you're going to cross it with, you're going to take this genotype, right? Because that's really the thing that helps you figure things out, is what alleles it has. So you're going to take these of the one you have, and you're going to cross it with another one. And again, it tells you what they are. For its tail, it's heterozygous. For eyes, it's homozygous recessive. For body, it's heterozygous. And for legs, it's homozygous dominant. Okay, so you're going to cross those. You want to write over here the alleles it has. So if it's heterozygous for the tail, what does that mean? What two letters? If it's homozygous recessive for the eyes, write the two alleles. Then you're going to take Punnett squares, that we've been using, and you're going to do a cross. Okay. So I'm going to do the first one just to show you. So this one, we're going to do the tail. So it's heterozygous, so we go back and see, because we're going to have to, it's heterozygous, the tail, if we go all the way back to here, the tail, straight and curly, and then it tells us here, tail, where is it? Curly tail is recessive, so it's a T, so the straight tail is dominant. It's heterozygous, so that's going to be the trait. For this new one, we're going to put it at the top, right? And then, what did you have for yours? We'll do that one too. So we had the curly tail, so it's homozygous recessive, so it's going to be like this and like that. So then we put that trait down here on our Punnett squares. Okay, then we're going to cross them. Put our alleles in. Okay, and then you just, right, because this letter goes there, that letter goes there for that one. This capital and these ones go together. This capital and that one goes together. So now, look at the directions. It says, so the possibilities of obtaining each trait. So this is your straight tail versus curly tail. And then it's heterozygous, but we know for the tail, we're just going to put S equals dominant. Okay, and curly is recessive. If you need to write this somewhere for yourself, do that. So then we have, anytime there's a dominant allele, we know we have the trait. So here we have a straight tail and a straight tail. So 2 out of 4, or 50% chance. Straight. What S? And then 2 out of 4 here. 2 out of 4, 50% curly. So there's your Punnett square. And then you do this for each trait. So then you would do the eyes, you would do the body, and you would do the legs. Show me a Punnett square. Show me the possibility of each phenotype. We're not going to worry about the genotype because some of those would be different. We're just going to say what is expressed. And if it has a dominant allele, that's the trait that's expressed. The only way the recessive could be expressed is 2 lowercase alleles. Okay, so that's that. And then you're going to answer this question. If you want to obtain offspring with all dominant traits, what would the genotype of the new organism need to be? So you want to think that through. So here, did we obtain all heterozygous? All dominant traits, right? So here 50% are recessive, curly. So if we wanted to obtain all dominant traits, which allele, two alleles does the one we cross have to have when we cross it with our original, right? So I want you to just see if you can figure that out. So here we tried this to homozygous recessive, we got 50% recessive. So we want, in the end, the answer to this question is, what's the cross for each trait that we have to have? Then we always get the dominant trait. So try that out. We'll discuss that in class. And then, so also it says, propose a way that student could obtain a new trait, like a body, okay, not seed in either parent. What would have to be different? Okay, so there's two answers to this. Think about how the gametes are produced, and maybe that could help. So it has something to do with gametes and how they're produced. Okay, that's one way you can get this. Go back and look at your notes if you're not sure. And then also, one of the words we used or learned today is mutation. That's another way. Maybe, and you need to explain how that works. I think I talked at the beginning. Okay. And then also, how might copper labs have determined which traits were dominant and which traits were recessive in student unhappiest? So again, look at that karyotype. That's probably going to be that one. Just explain that a little bit more. And then propose a new trait and give me the alleles and dominant and recessive genotypes for those. And then we'll discuss this in class. I'm going to add the karyotype lab as well, where you just go to the online thing, complete the lab, and turn in the screenshot when you're done. So this is kind of how you do that lab. Let me know if you have any questions. Go ahead and complete this. I'll give you one more assignment. We're going to do some probability stuff as well, because there's a way we can figure out the possibility of all these traits occurring together. That's kind of our next exercise, a dihybrid cross. And we don't have to necessarily do as much work. It's a little bit easier. But here we're just practicing Punnett squares. So let me know if you have any questions. You can use the website, the Google Classroom, to ask me questions, or you can email me, whatever works.